In the name of Allah, who is most beneficent and merciful. My name is Sumaya. My ID is 014. I'm from Chemistry Department. Today, the topic of my presentation is synthesis and reactions of lactic acid and formic acid. So first of all, you can see contents of my presentation, which include lactic acid and formic acid. In lactic acid, I'll discuss introduction and basic information, its synthesis by chemical means and by fermentation, then its biological reactions. After that, I'll discuss about introduction of formic acid and its basic information, its synthesis in industries by fermentation, and at last, biosynthesis of formic acid. So let's start with the introduction of lactic acid. A colorless or yellowish syrup, water-soluble liquid, having molecular formula C3H6O3, produced during muscle contraction as a product of anaerobic glucose metabolism, abundant in sour milk, prepared usually by fermentation of cornstarch, molasses, potatoes, etc. are synthesized. It is used chiefly in dyeing and textile printing, as a flavoring agent in food, and is also used in medicine. After that, you can see basic information about chemistry of lactic acid. The structure you can see, molecular formula I have told you already, and molecular weight is 90.08 of the lactic acid. Now we'll discuss about synthesis of lactic acid. So there are main two processes of synthesis. One is by chemical means and the second one is by fermentation. But first of all, we'll discuss production by chemical means of lactic acid. Lactic acid production by chemical synthesis using the Lacto-Nitrile, which is byproduct of acrylonitrile technology. First of all, hydrogen cyanide is added to liquid acetaldehyde in the presence of a base catalyst and a high pressure. When lacto-nitrile is produced, in this equation you can see when hydrogen cyanide is added to the acetaldehyde, it gives us lacto-nitrile. And in next step, when it is hydrolyzed with the help of the water and sulfuric acid, it gives us lactic acid and ammonium salt. So what happens in next step? The lactic acid is esterified with methanol and the ethyl lactate formed is recovered, purified by distillation and hydrolyzed with acidified water to produce lactic acid and methanol. In this reaction, you can see lactate, which is formed in the previous step. It is being hydrolyzed with the help of the acidified water, and as a result, we get lactic acid and methanol. At last, methanol is separated by distillation. Here you can see reason of using lacto-nitrile for chemical synthesis of lactic acid. As we know, there are many possible routes for the production of lactic acid by chemical synthesis. But why we use lacto-nitrile route? Because none of these routes are technically and economically feasible except for the routes that use lacto-nitrile as the raw material. Then we'll discuss about fermentive synthesis of the lactic acid. So there are many pathways in fermentation of the lactic acid. First one is homofermentive process. In homofermentive process, basically homofermentive bacteria are used which convert glucose to two molecules of lactate and use this reaction to perform substrate level phosphorylation to make two molecules of ATP. In reaction, you can see when glucose, two molecules of ADP, adenosine diphosphate, and two molecules of inorganic phosphate react, then we obtain two lactate molecules and two molecules of adenosine triphosphate or ATP. Then there is heterofermentative process. In this process, bacteria produce less lactate and less ATP, but produce several other products. What they produce? Other products, ethanol and carbon dioxide. In this equation, you can see glucose, ADP, and inorganic phosphate react and give lactate, ATP, with byproduct of ethanol and carbon dioxide. Third one is Bifidum Pathway. Bifidum utilizes a lactic acid fermentation pathway that produces more ATP than either homolactic fermentation or heterolactic fermentation. In these two reactions, you can see there were never 5 molecules of lactate, but in this reaction, you can see 5 lactate molecules are obtained. It means it produces more ATP than either homolactic fermentation or heterolactic fermentation because it is producing more lactate. Then we will discuss about applications of lactic acid fermentation. Lactic acid fermentation is used in many areas of the world to produce foods that can't be produced through other methods. The most commercially important genus of lactic acid fermenting bacteria is Lactobacillus, though other bacteria and even yeast are sometimes used. The most common applications of lactic acid fermentation are given. First one is fermentation, second one is sorbet, and third one is yogurt. The main method of producing yogurt is through the lactic acid fermentation of the milk with harmless bacteria. The primary bacteria used are typically Lactobacillus and Streptococcus. So these bacteria produce lactic acid in the milk culture, decreasing its pH. The bacteria also produce compounds that give yogurt its distinctive color. Then we see another application of lactic acid in vegetables. Lactic acid bacteria already exist as part of the natural flora in most vegetables. Lettuce and cabbage were examined to determine the types of lactic acid bacteria that exist in the leaves. Now we'll talk about biological reactions of lactic acid. So first one is how it is produced during exercise. During power exercises such as sprinting, when the rate of demand for energy is high, glucose is broken down and oxidized to pyruvate. And lactate is then produced from the pyruvate faster than the body can process it, causing lactate concentrations to rise. The production of lactate is beneficial for NAD regeneration, which is used up in oxidation of glyceraldehyde 3-phosphate during production of pyruvate from glucose. And this reaction makes ensure that energy production is maintained, and as a result, exercise can continue. During intense exercise, the respiratory chain cannot keep up with the amount of hydrogen ions that join from NADH and can't regenerate NAD quickly enough. In this reaction, you can see glucose is being converted in the pyruvate with the help of the oxidation, and pyruvate is being converted in the lactate with the help of the fermentation. And in the whole process, NAD and NADH are being converted into each other. So resulting lactate in this reaction can be used in 2 ways. First is, it is then directly used to fuel in the Krebs cycle. The second one is, if blood glucose concentrations are high, the glucose can be used to build up the liver's glycogen stores. Then we see biological importance of lactic acid in neural tissue energy source. Although glucose is usually assumed to be the main energy source for living tissues, there are a few reports that indicate that it is lactate and not glucose that is preferentially metabolized by neurons in the brain of several mammalian species. Non-primary source needed according to the lactate shuttle hypothesis is the lactic acid. So glial cells are responsible for transforming glucose into lactate and for providing lactate to the neurons. So it is important in neural tissue energy source. Here you can see there are units to measure lactic acid in the blood, and there are 2 parameters, venous and arterial lactate. These are the units in which venous lactate and arterial lactates are measured. Blood tests for lactate are performed to determine the status of the acid-base homeostasis in the body. Blood sampling for the purpose is often arterial because lactate levels differ substantially between arterial and venous, and the arterial level is more representative for this purpose. Moving towards, we will focus upon second topic of our presentation, which is formic acid. So first of all, I'll introduce formic acid. Formic acid is composed of a carboxyl group attached to a hydrogen atom. IUPAC name is methanoic acid. Molar mass is 46 gram per mole and boiling point is about 100.8 degrees Celsius. Here you can see basic information about chemistry of formic acid. Structure, you can see molecular formula is CH2O2. Molecular weight is 46.025. Now we'll talk about its synthesis. In industries, it can be synthesized by chemical means. So first of all, by methyl formate and formamide. When methanol and carbon monoxide are combined in presence of a strong base, the result is methyl formate. And you can see in this reaction, methyl formate is formed as a result of methanol and carbon monoxide reaction. In industry, this reaction is performed in the liquid phase at elevated pressure. Typical reaction conditions are 80 degrees Celsius temperature and 40 atm pressure. Most widely used base is sodium methoxide. Hydrolysis of methyl formate produces formic acid. Like you can see, when hydrogen and oxygen like water is added to the methyl formate, then it gives us formic acid with methanol. Then what happens in next step? So efficiently hydrolysis of methyl formate requires a large excess of water. Some routes proceed indirectly by first treating the methyl formate with ammonia. As you can see in this reaction, methyl formate is being treated with ammonia and it gives us formamide, and which is then hydrolyzed with the help of the water and with the help of the sulfuric acid, and it gives us formation of enzyme. A disadvantage of this approach is the need to dispose of the ammonium sulfate byproduct. This problem has led some manufacturers to develop energy efficient methods of separating formic acid from the excess water used in direct hydrolysis. So in one of the processes used by BASF, the formic acid is removed from the water by liquid-liquid extraction with an organic base. So the second process by chemical means is by hydrogenation of carbon dioxide. So simply hydrogen is added in carbon dioxide and we get as a result lactic acid. Then how it is synthesized in laboratories? By steam distillation. In the laboratory, formic acid can be obtained by heating oxalic acid in glycerol and extraction by steam distillation. Glycerol acts as a catalyst as the reaction proceeds through a glyceryl oxalate intermediate. If the reaction mixture is heated to high temperature, allyl alcohol results. So the net reaction is given as it is obtained by heating oxalic acid in glycerol. In this reaction, you can see carbon dioxide is also obtained as byproduct. So another illustrative method in laboratories involves the reaction between lead formate and hydrogen sulfide. In this reaction, you can see lead formate and hydrogen sulfide react to form the lead sulfide. But we also get formation of acid. The third process which takes place in industries is electrochemical production. So it has been reported that formate can be formed by the electrochemical reduction of carbon dioxide at a lead cathode at pH of 8.6. And the reaction you can see when carbon dioxide and water react and there are addition of 2 electrons, we get bicarbonate ions and hydroxyl ions. And if the feed is carbon dioxide and oxygen is evolved at the anode, then the total reaction will be carbon dioxide and collection of carbon dioxide and hydroxyl ions, and as a result we will get bicarbonate ions and oxygen. This has been proposed as a large-scale source of formate by various groups. The formate could be used as feed to modified E. coli bacteria for producing biomass. So these were all the processes by which we can produce the formic acid in the laboratories in industries. So now I'll discuss about biosynthesis of formic acid. It is named after ants which have high concentrations of the compound in their venom. In ants, formic acid is derived from serine through a 5,10-methyl methanol tetrahydrofolate intermediate. So this is the structure of the serine. The second process of the biosynthesis of formic acid is by glycine. When we add oxygen in glycine, simply we get formic acid with carbon dioxide and ammonia. And these are the references from which I have collected all the material of my presentation. That's all from my side. Thank you.